How are you doing? Right, we're going to talk about the histology of the ovary. Just come from a 3-hour revision session with Year 2 medical students who know a lot, which is great, and they really challenge me, which is great but tiring. But the ovary, okay, so the ovary is the organ, the gonadal organ, in which we find oocytes, egg cells, and one of those oocytes is going to be released each month in humans, ready for fertilization by spermatozoa to create a new individual. So we're going to have a look inside the ovary, and what we're going to see is we're going to see lots of oocytes, and some of them are going to be kind of in a resting state, and we're going to see oocytes surrounded by supporting cells, follicular cells, which will become granulosa cells. There's going to be a lot of words. And we'll see how that follicle, so the cells that are supporting the oocyte form a follicle, like a ball of cells around the oocyte. We'll look at how that follicle develops and gets that oocyte ready to be released during ovulation. So we'll see lots of circles, I think. Okay, so we should clean the slide. We should look at the overall structure of the ovary first. Will only take a minute, and then we'll dive in and see what we can see and work our way through the timeline of the oocyte, right? Is that in focus already? Wow. Okay. That's my lowest power. This is quite a big, this is the whole ovary. Told you, lots of circles. Following the outer edge there. And there's not a great deal of stain here. So what we need to do is identify all of these circles and recognize these, oh, this looks perfect. Recognize these patterns. Got a bit of yellow staining, a little bit of purple staining, not a lot of staining. So the ovary, like most organs, has a cortex and a medulla. The cortex is on the outside. That's what we're looking at there. We're up against the edge. Oh, fun. If I zoom in, this is my, what's that, my 10 times objective. Fun fact, maybe, is do you see how on the surface of the ovary there, look at that, there's a single layer of cells. Isn't that interesting? That gets called the germinal epithelium. It's not really a germinal epithelium at all. I guess it's where scientists originally thought that oocytes came from. It's not a germinal epithelium, these aren't germ cells, but this is a single layer of cells covering the surface of the ovary that gets called the germinal epithelium. So watch out for that. That's a point of confusion. And if the germinal epithelium is the outermost layer of cells, just deep to the germinal epithelium, we can see a connective tissue layer. This is the tunica albuginea, just as we saw in the testis. So this is the connective tissue that is supporting the ovary, giving it shape. You know, it'll move into the ovary. The ovary's got more connective tissue inside it. So that's the tunica albuginea. So those are the outer two layers, and look, we're starting to see circles right up against the tunica albuginea, but that's for in a moment's time. Right, let's go back out to my lowest power. And in terms of cortex and medulla then, in the cortex, that's where we're seeing these interesting circles. And where the cortex becomes the medulla isn't particularly clear, but as we move towards the centre of the organ, well, we're seeing some specific things here, but right, there's one edge, there's the other edge, so in there's the middle-ish, that's the medulla in there, and what we're seeing in the medulla mostly will be blood vessels, lymphatics, nerves, that sort of thing, right? We're not going to look at this again later, so if I just pop up to a higher power, this is what the medulla looks like. It's okay, there's a lot of circles in the body, forgive me. But the circles we're seeing here are just your bog standard blood vessels and what have you, right? So that's the medulla and the cortex. On to the interesting stuff. All right, so what's going on in here? What are we looking at? That's quite a nice spot. Well, just like when we were looking at the testes and producing spermatozoa, the aim of the game is the same. We're trying to produce a cell with a haploid arrangement of chromosomes. So what you want to do is you want to have your mother's chromosomes and your father's chromosomes and put them together and create a new genetic individual with the correct number of chromosomes. So the oocytes here, the final form needs to contain a haploid set of chromosomes to meet with a haploid set of chromosomes in the spermatozoa to form a new genetic individual with a diploid set of chromosomes. So the cell division process that we're looking at is meiosis. I talked a bit about this when we looked at the testis. I might have done an okay job, I'm not sure. Mitosis is your general cell division that's happening throughout the body in terms of cell repair and making new cells and growth and that sort of thing. Meiosis is different because meiosis also jiggles around the DNA, so you've got just the right amount of DNA at the end. You've got all the genes but a haploid set. So the difference between the testes and the ovaries is that all of the oocytes were created during the embryonic and the fetal periods. So in the ovary, all the oocytes here were created during the embryonic and fetal periods. So in the human, at around the age of puberty, when puberty starts, there are about 500,000 oocytes, and that's it. That's the lot. And that's plenty, to be honest. Whereas in the testes with spermatozoa, constantly new spermatozoa are being created. That's not what's happening here. And when these oocytes were being created, they started the first stage of meiosis. So meiosis has 2 cell divisions. They started the first meiosis division, but they didn't complete it. And then they paused, and now they're here. And we're going to watch them move into the second stage of meiosis. Alright, so let's zoom in, and what we should see, if we get out to that tunica albuginea level, well, look at that, we can see there, we can see lots of circles, and I don't know if you can imagine this, but we can see circles in slightly different stages of development. This is perfect. We've got some little circles and some slightly bigger circles and then some slightly more complicated circles and then some even bigger circles again. All right, so if I zoom in, this is my 20 times objective. And we can see some large cells there. So with a central nucleus, quite a lot of cytoplasm around the outside, a bit like a fried egg. And you might imagine that there are some squamous cells, some very thin, flat cells wrapping around that. So that is the primary oocyte, and that is a primordial follicle. So the cells surrounding it are follicular cells, and that's the simplest form. So that there are many, many primordial follicles, and this is that primary oocyte fixed in that first stage of meiosis. So there are lots of these primordial follicles around, right? And they're around the edge. And then we can see, as we're moving kind of towards the middle of the ovary, we can see there's one there as well. That's quite a good one. We can see that the follicle has started to develop. So those follicular cells have become a single layer of cuboidal cells. These follicular cells are going to become granulosa cells. They're going to become cells that make estrogen and make progesterone and have a role in the menstrual cycle and pregnancy and that sort of thing. And in the middle there, we have that primary oocyte. So it's still a primary oocyte and it's getting bigger. And the follicular cells are supporting that growth. The follicular cells are also going to have receptors for follicle stimulating hormone, FSH. The follicle stimulating hormone is stimulating the cells of the follicle and stimulating this growth. So we see that the first part of the primary follicle is that, where we start to get a single layer of cuboidal cells. And we can see some other cells here, or some other follicles that are maybe a little bit further along with 2 layers of cuboidal cells. And as we go up here, this is a slightly more developed primary follicle. And now we've got multiple layers of follicular cells, granulosa cells. That oocyte in the middle has become much larger. And can you see there's a ring of cells running around the outside of those granulosa cells on the outside of the follicle. So those are cells from the stroma, so the cells of the tissue of the ovary. Those cells are becoming thecal cells. Now, thecal means like a sheath or a box. So these are the cells around the outside, and these are going to be cells that support the follicle as it grows. So that's still, I would say, a primary follicle. And look, if we look over here, we're seeing even more development. We're seeing multiple layers of granulosa cells. We're seeing the follicle in the middle, and we're seeing maybe there, so the thecal cells will form 2 layers, a theca interna, so an internal covering, and a theca externa, an outer covering. And they're going to do slightly different things. All right, now, if we look, I'm wondering if we look closely. Well, actually, we can see there, can't we? I'm looking for the zona pellucida. The zona pellucida, it's a glycoprotein. That's fancy, isn't it? Sugar and protein. It's a glycoprotein covering to the oocyte, which essentially its job is to ensure that only one spermatozoon will fertilize it and no others will. And that develops kind of... The primary follicle is going to become a secondary follicle, and okay, that's pretty good. Can you see there's the oocyte in the middle, and it's got a yellow ring round it, so it's now surrounded by the zona pellucida that's formed. And how well can we see? So the theca interna and the theca externa layers. Well, the theca externa layer is kind of going to be a connective tissue support. The theca interna is going to produce androgens that the granulosa cells will use to make estrogen. But can you see here in this follicle, this is in the next stage. So look, it started to change here, isn't it? The granulosa cells have started to pull in fluid. So those thecal cells are making some fluid, and the granulosa cells are pulling in the fluid. Now we're seeing spaces in between the granulosa cells. We're starting to see a space forming. This space is going to form a single space, and that's going to be the antrum. So it's through these processes by which the follicle changes that the primary follicle becomes the secondary follicle. So as we start to see fluid being pulled in and an antrum forming, then we start to think of this as a secondary follicle, but it's still a primary oocyte. Now, let's see if we can find the next stage, which will be that fluid filled cavity becoming a single cavity, right? So that's what we're seeing there. But I don't think that's a very nice shape. That's just the plane of section has caused that. And then as it pulls more fluid into it, it starts to look, oh, there we go. Starts to look like that. That looks like an eye, doesn't it? So we're starting to see spaces forming in there. Those spaces are supposed to be in there. That's part of the development of a secondary follicle. And there we go. Oh, this is great. See, that's quite nice, isn't it? there's now a space in there. So we've got that primary oocyte in the middle, which is getting bigger and bigger. We've got that space forming around it in between the granulosa cells. We've got the theca interna and the theca externa around it. That's our secondary follicle. And it's almost there. It's almost ready. But it needs to become a Graafian follicle or a mature follicle. Oh look, there's the next stage. So as you can see, there are lots of follicles going through this process at once. This isn't a human ovary, so sure, it's a little bit different because some animals, of course, give birth to multiple offspring at once, whereas humans just tend to do one at a time. So look, there's that follicle continuing to develop, and there's quite a few changes there. Now what I'm looking for is where the, that's, it's almost like I scripted this. Look, now the oocyte is there in the middle, surrounded by some supporting cells, and you've got the cells around the outside. It's actually still attached to the granulosa cells, but there's what we call a heap of cells, a cumulus oophorus, which is Latin for a heap of like egg supporting cells, something like that. Cumulus means a heap, a pile. But we just can't see it in that plane of section. But that, now we're starting to look at a Graafian follicle. Let's keep going, see if we can find the heap. So like these circles, which haven't got an oocyte in them, that's just, they're just missing them from the plane of section, because of course this is a big sphere, right? I'll bet I won't find one, because everything else has been going really well, isn't it? No. You might have to use your imagination. Maybe that's, I don't know, maybe that's as close as we're going to get. But in the Graafian follicle then, we have our oocyte. It has its zona pellucida. It has a space around it, an antrum around it, and then we have the granulosa cells, and then we have the fecal cells. And here, the oocyte has continued through meiosis, and it's gone through meiosis 1, and it's chucked off a polar body, some extra stuff it doesn't need, and it's passing into meiosis 2. But it's going to pause in meiosis 2 for ovulation, and it'll only complete meiosis 2 when it meets a spermatozoon and fertilization begins. All right? So if that's our very pretty secondary follicle, all of these circles in here, they're just missing the oocyte because of the plane of section. These are our Graafian follicles. So they've got a big space inside them, and it's from the Graafian follicle that the oocyte will be expelled during ovulation, and the oocyte will pass into the fallopian tube, the uterine tube, and it can sit there for probably a couple of days, and spermatozoa can move in, and fertilization, right? So Graafian, that's why that gets named a Graafian follicle, and so on. So if I zoom back out again to give you a sense of scale, so this is back to my 4 times objective, 10 times to my eyes, so 40 times magnification for me. This is the elephant in the room. This huge great big space, this huge great big thing in the middle that I haven't talked about. This is the corpus luteum. This is what's left after the oocyte is ovulated. What on earth are we looking at here? Well, there have been quite some changes. So you can see that the granulosa cells have changed. So the corpus luteum, luteum refers to yellow in Latin. So in the ovary, this looks like a yellow body. So corpus, body, lutein, yellow. So this is a yellow body. Those granulosa cells have become, well, they look quite different now, don't they? So the granulosa cells have become granulosa lutein cells, and the theca cells have become thecal lutein cells. And these granulosa cells are going to produce progesterone, which is why they look like this. And the stroma and blood vessels have moved in here as well. And that progesterone is going to signal to the endometrium to hang around for a while, just in case fertilization happens, so that the embryo can implant into the endometrium in the uterus and pregnancy can continue. There's also some estrogen being made which will inhibit follicle stimulating hormone for a little while. It's part of the hormone dance that makes the menstrual cycle, right? So we've got multiple corpora luteum in this tissue, so I don't know what animal this has come from. But look how big it is compared to everything else. Now, the key idea here is that we can see that at any one time, many primordial follicles are developing and becoming primary follicles, maybe secondary follicles and Graafian follicles, but every month only one is going to make it. Only one is going to be selected for ovulation in humans anyway. And the rest will all regress, atrophy, and be broken down and be reabsorbed, including the oocyte. It's gone. It's had its chance, it's failed, it's gone. And the selection mechanism, as far as I understand, the selection mechanism by which that oocyte has been picked as this month's winner, it's not entirely clear how that works. But it seems to be important in selecting the best oocyte, the best cell for fertilization, right? It's improving the chances of fertilization, making it as likely to happen as possible, and making a new person. But only one of all of these cells that are going through these processes will be selected each month, and everything else will be removed. So 500,000 follicles, 500,000 oocytes is still plenty, but it, in fact, so if you think the reproductive age extends to about 40 years old, maybe, damage to DNA over that time can cause some problems. But mathematically, there should still be plenty of oocytes left, but changes occur. All right, so that is the histology of the ovary. Again, I think it's quite different to all the other tissues. It looks quite weird, but if you start at the outside and you start seeing all those circles, you recognize all those primordial follicles that kind of sat there waiting for their chance. And then you can see, as you move away from the edge, you see those developmental stages of the primary follicle developing and getting more follicular cells and more layers, and the oocyte itself is getting bigger, and then you get those thecal layers forming, and then it becomes the secondary follicle as it starts to pull in fluid, and then as it pulls in fluid it makes a space around the oocyte, and the oocyte remains attached by the cumulus oophorus, and that oocyte is progressing into the second phase of second meiosis, and now you've got the Graafian follicle, and then one of those will be picked and the oocyte released during ovulation. And we can see, it's very handy actually, laying all this out in front of us, isn't it? But you can see those processes happening in the ovary. Right, it's so big, look, I can't fit much of it in. Cool. All right, that was good to look at. See you next week.